I am delighted to introduce the first speaker of the session today, Professor David Harlan. David is currently the William and Doris Nutt Professor of Medicine at the University of Massachusetts School of Medicine, and he's also the co-director of the Diabetes Center of Excellence there. And he's had an illustrious research career, which started some time back, where he was based at the NIDK of NIH Intramural Research Program, and he headed up the departments of diabetes and endocrinology. And I'm particularly excited to hear David speak this morning because almost a year ago, I had the pleasure of hearing him speak at the Levine meeting at the City of Hope. And he gave an absolutely wonderful after dinner talk on the history of insulin that I found inspiring and motivated me to buy many books and to improve my understanding of the history of this amazing hormone. So without further ado, let me hand over to David to get things started.
Thank you, Anna. Well, as Sung and Anna implied, I devoted my professional life to understanding diabetes some 35 years ago. And the major theme that I want to strike for you all today is the noble heritage that we in the diabetes field enjoy. Not only noble, but I say often Nobel, and you'll see why. I very much appreciate this opportunity to share my passion with you all today, and I'm thrilled that so many people share that passion. I'll be describing several major themes as I recount the history of insulin. They are good ideas versus brilliance. The role that serendipity plays in science, the very important role of persistence in all of scientific advances, and the often unforeseen consequences of scientific pursuits. I'll describe insulin's initial purification and use, and notice I don't use the word discovery. It's been around for millennia, but its initial purification and use. I'll describe the role that insulin played in the whole field of endocrinology and our ability to measure hormones. And then the role that it played in the molecular biology revolution that leads our biotechnology industry today. I'll describe how once insulin was isolated and cloned and created in bacteria, it's been modified for improved diabetes therapy. And I'll describe the true nature of scientific achievement, including often the acrimony that's associated with success. So I do have some disclosures. One is I am extremely persistent. Once I pick a goal, I focus on it laser-like. And as I said, 35 years ago for me, that was understanding type 1 diabetes sufficiently well so that we might see the disease eradicated. I disclose that I have TB, by which I mean true believer. I am convinced that we can create a healthcare system worthy of that name, and for those with diabetes, a cure can and will be found. And I do have a serious disclosure that I have co-founded a company called Stability Health. The company's focused on improving the way diabetes care is delivered throughout this country. So let's start from the beginning. Diabetes is an illness that's well documented in the medical literature. It has a longer history than virtually any other illness that we manage today. There's descriptions of diabetes going back 2,000 years. I include an early description there from Eritreus. Diabetes is a meltdown of the flesh and limbs into urine. Its course is of a cold nature. The patients never stop making water, as if the opening of aqueducts. The nature of the disease is chronic and it takes a long period to form, but if the constitution of the disease be completely established, the melting is rapid and the death speedy. And as we'll see in the prognosis of this disease, the course that Eritreus described 2,000 years ago was the course right up until the beginning of the 20th century in 1912, when Dr. Frederick Allen, shown there in the rather stern picture, came up with a treatment that was basically carb restriction, severe carb restriction. The therapy relied on what he called thrice boiled vegetables to eliminate all the simple carbs. And then Elliot Joslin came along as the first true scientist trying to understand diabetes pathophysiology and develop better treatments for patients through education. So as I've mentioned, the life expectancy for diabetes through the millennia was dismal. If once people were diagnosed with the disease, they could live months, maybe a year, until Frederick Allen came along. And then people, as you'll see in a few minutes, were surviving a year to 3 years with this carbohydrate poor starvation diet. And that's when this man came along. Let me say a few words about Banting. He was born outside of Toronto and started his medical school at the University of Toronto, but that training was cut short by World War I. He served honorably in the Canadian Army in France and was awarded the Military Cross for heroism under fire. He was grievously injured. He had a laceration of his arm, but refused to leave the operating suite until he passed out. After the war, he returned to set up a general practice in London, Ontario, and nobody came. And when I say nobody, I visited the home where he set up this practice, and I'll show you a picture in a moment. But in the first month that he opened the practice, he had one patient, and that was another returning soldier who wanted a prescription for alcohol because it was during the Prohibition period, and the only way you could have alcohol is with a doctor's prescription. Anyway, Banting was looking for some way to make some money, and a new medical school had opened up in Western Ontario, and he volunteered to teach an anatomy lecture there, and they assigned him the pancreas. So he had to learn pancreatic anatomy, and then he learned that the internal secretion, as it was called at the time, it was known to be made in the pancreas based on von Behring work from decades earlier. But it was subsequently thought that the internal secretion was not just made in the pancreas, but within the pancreatic islets within the pancreas. So Banting gave his lecture, and by all accounts, it was a bad lecture. He was never known to be a good public speaker. Then he went back to his empty office, and as chance would have it, here's the first bit of serendipity. That day, he received his copy of the Journal of Surgery, Gynecology, and Obstetrics, and in it was an article by Dr. Moses Barron. And Moses Barron begins his article saying, any reference to the pancreas is secreting a hormone necessary for the utilization of sugar by the tissues is misleading, as that function is, accurately speaking, exercised by only a very small portion of the organ called the islets. Well, Barron goes on to describe that when humans with chronic pancreatitis die and are autopsied, or if you take an animal and ligate its pancreatic duct such that the animal develops pancreatitis, and you let that animal survive from that surgery, if you look a few months later, what's left of the pancreas is a nubbin of what it originally was, but highly enriched in these islets with very little of the pancreatic acinar tissue remaining. So Banting read that and he went to sleep. And then he woke up in the middle of the night at 2 a.m. and he had a diary by the side of his bed, and he wrote in it, you can see on the right here his actual words, but I've transcribed it here. First, you notice he was not a good speller, because this is how he spelled diabetes. But he said, ligate pancreatic ducts of dog, keep dogs alive till acini degenerate, leaving islets. Try to isolate the internal secretion of these to relieve glycosuria, and he's misspelled glycosuria too. So that was the inspiration. And this, if you visit Western Ontario, the house is still there, and it's a historical site. And if you identify yourself as a diabetes geek like I am, they'll let you sit on the bed. This is the bed. The clock is set for 2 a.m. This is where the whole idea came from. So Banting went to University of Toronto, where, as chance would have it, one of the world's authorities on carbohydrate metabolism, J.J.R. McLeod, was the professor. And Banting described his idea to him, and McLeod said, first, what gives you the idea that you'll be able to purify the internal secretion when the best scientists in the world have tried to do this without success? But it's an intriguing idea. I like your idea of ligating the pancreatic duct. I'll see if I can find a student that'll work with you for free, and you have to work for free. Banting did this for free. And let's see if you can gain any headway. So Charlie Best was the student that signed up to work with Banting, and as Sung alluded to, they spent a warm summer in 1921 doing the experiment that Banting conceived of on that October 31st night. And they obtained their first positive results on July 30th, 1921, just 7 months after Banting had his middle of the night dream. They had pancreatectomized a dog, so it was dying of hyperglycemia and ketosis, and they injected some of the extract from another dog's pancreas. And when they came back a few hours later, the dog was standing up and wagging its tail, and story tells it that Banting fell to his knees, knowing that they were onto something. Later, when McLeod saw the results of their experiment, he pulled in James Bertram Collop, who was a trained biochemist, to help them purify insulin. Such that a mere 13, 14 months later, when Leonard Thompson, born in 1908 and diagnosed with diabetes in 1919 when he was 11 years old, when he was admitted to the hospital in December 1921, he had been treated with the Allen diet, and you can say as a 13-year-old he weighed 65 pounds, much of which was edema, they say, and he was ketotic. Here's an actual copy of his hospital notes shared by Professor Bernice Zinman with me a few years ago. And I'm just transcribing here what's written, that he was on December 14th feeling well and drinking fluids freely. But on January 11th, and this is the birth date that we all celebrate, he got 15 cc's of what was called McLeod's serum into each buttock, 7.5 into each buttock. And the result was an infection at those sites, or at least an area of induration, but not much benefit. Then they used Collop's crystallized, purified form of insulin, and Thompson's blood glucose came down. He recovered, his ketosis disappeared, and the rest of the story is that he survived an additional 14 years, which was unheard of back then. People did not recover from diabetic ketoacidosis. This story spread through the world like wildfire, as you might imagine. This is a newspaper clip from the Toronto newspapers. Have they robbed diabetes of its terrors? Here's Banting, Best, J.J.R. McLeod, and Collop. They used Collop's fractional precipitation purification method using acid alcohol, and paradoxically, the whole idea of having to ligate the pancreatic duct went away with this. You could take a whole pancreas and get urine out with just the acid alcohol extraction. They worked with Connaught Laboratories to figure out large-scale production with other scientists. I've named some here to further purify insulin. And then once the purification scheme was mature, a diabetes clinic was established at Christie Street Hospital. Now, you might think that these four would be extraordinarily happy for each other and celebrate the advance. Oh, this is just a picture I took from Eli Lilly showing the large-scale purification of insulin that they developed by having cow and pig pancreases shipped to them. But in fact, there was tremendous infighting, and I showed you the picture of me visiting the Banting home. The curator of that home was visited by one of Best's sons years later. And the curator said, "What does your father think was his greatest contribution to the discovery of insulin?" And Best's son said, "Well, Dad always said it was pulling Fred off of Collop when he was strangling him, because they were fighting in the lab about who deserved credit for the purification of insulin." It really became quite acrimonious. Dr. Frederick Allen, scientists like Allen and Joslin immediately wanted access to Dr. Banting's insulin. And this is a letter that Dr. Banting received from Frederick Allen in August 16th of 1922. He said, "I take pleasure in informing you that our first results with your pancreatic extract have been marvelously good. You deserve all congratulations, but as you will receive them so abundantly, I need not express myself at length now." And Allen, of course, was right. Banting and MacLeod, not Banting and Best, were awarded the Nobel Prize on February 26, 1923. And to this day, it's the most rapid awarding of a Nobel Prize after the announcement of a scientific advance. It was the shot heard around the world. And for those of you who haven't read Banting's Nobel lecture, I strongly encourage that you do so, because in it he acknowledges the important role that the purification efforts by the companies and Collip played in insulin's use. He describes how insulin was variably sensitive in different individuals. He describes hypoglycemia reactions. He says that some people develop hypoglycemia symptoms at different blood sugars. And he concludes his lecture with this quote, "Insulin is not a cure for diabetes, it's a treatment." And I'll come back to that later. So people remember stories, and I'd like to include this one. One of his early famous patients was Elizabeth Hughes. Here she is with her father, and for the young people, they might not know who Charles Evans Hughes was, but here's his picture in 1916 when he was running for president against Woodrow Wilson in what was up to that point one of the closest presidential elections in American history. He had previously been the governor of New York. After 1916, he became an associate justice of the Supreme Court and then subsequently our Secretary of State and ultimately the Chief Justice of the Supreme Court. So clearly a man who was well-connected, wealthy, and educated. And here he is with Elizabeth in 1916. She was born in 1907. She was diagnosed with diabetes in 1919 when she was 11 years old. And as a typical 11-year-old, weighed about 75 pounds. And because they were rich and influential, they were able to get Dr. Allen to be her doctor. And he treated her as he treated all, such that by the time she was 15 years old in 1922, she was alive. But as a 15-year-old, she weighed 45 pounds. When they got word of Banting's success in Toronto, they were able to get her there, and she became one of Banting's early treatment patients. And when she got her first shot of insulin in August 1922, she wrote in her diary that insulin was simply too wonderful for words. Within 4 months, she weighed 105 pounds, kind of a normal weight for someone that's 5 feet tall. And the rest of the story is that she went on to graduate from Barnard College. She married, she had 3 children, and she died at age 74 in 1981, never telling many of her closest friends that she had diabetes. She was embarrassed. It was something that she didn't want known about her. Now, I told this story to one of my patients who my family all came to know. This is a lovely woman named Amelia Frank. This is her on her 100th birthday. But I told her this story one time and she said, "Well, that's odd." She had gone to Barnard. She said, "I was friends with an Elizabeth Hughes." Turns out Amelia and Elizabeth went to college together and were friends. And Amelia never knew that Elizabeth had diabetes. So anyway, overnight, the prognosis for those with diabetes improved such that individuals were now living decades after their diabetes was diagnosed. And then over the next 20 years or so, various advancements were made in insulin purification and in extending the duration of insulin's action so that people didn't need to take 4 shots of insulin a day or 3 or 4 shots of insulin a day. Now I'm going to shift to some other science based on the study of insulin, 3 of which have been recognized with Nobel Prizes. One is by Rosalind Yalow. She focused first on the insulin assay, and from that developed radioimmunoassays, which revolutionized endocrinology. Dorothy Crawford Hodgkin, who was inspired to pursue X-ray crystallography when she first saw crystals of insulin. And Frederick Sanger, who won the Nobel Prize for the ability to determine the amino acid sequence of proteins, and the first protein that he did that for was insulin. And he later developed techniques to sequence DNA and RNA, as you'll hear in a moment. So the Nobel Committee disagreed with Fred not being very academically brilliant because they gave him the Nobel Prize twice. He's one of the few scientists in the history of such awards to win 2 Nobel Prizes, one in 1958 for the proteins that I've mentioned, and again in 1980, along with Wally Gilbert and Alan Maxam, for contributions concerning the determination of a base sequence in nucleic acids. Now, speaking of Dr. Gilbert, I had the privilege of sitting with him a little over a year ago, and I'm going to play several clips of my interview with him.
You are quoted as saying, more or less, it takes as much effort to answer a small question as a big one. So then don't waste your time on small questions.
Yes, I do say things like that.
Yeah. But I wonder if there's room for that in science anymore.
Well, there is in places.
Yes.
It is one of the problems that so much of the science has become big science. It is then hard for people to function in the way which we could. So when I began working in messenger RNA, when I started biology, I worked on messenger RNA, which is a fundamental discovery. We worked on ribosomes, we worked on mechanisms of protein synthesis, discovered certain interesting facts about how you made proteins. And then I went off and worked on the repressor. And each of these has become gigantic fields of science and full of fantastic detail. But the amazing thing is, people make totally novel new discoveries in science all the time. And when those fields are new and novel, then they're simple and they're easy to do, and you can do something interesting.
Yeah.
We think of humans as having 20,000 to 30,000 protein genes, plus another many thousand small RNA genes and other things floating around, and all sorts of RNA molecules floating around, most of which are of unknown function. And each of these genes is modified post-translationally in many, many ways, so that all of those modified by alternative splicing, modified by chemical modification. Complicated story, fantastically complicated.
Yeah.
And it's surprising that people can find totally new things in that, but they do.
Yeah.
So there are totally new things coming out in science.
So lesson number 1 for you all is, for you young people in particular, when you start down a scientific project, make sure that question is worthy of your time and effort.
What would you like your legacy in all of this effort to be?
Well, my legacy is actually in the DNA sequencing, and so which is of course now bypassed all of that.
Yeah, right.
The fun part of science is that everything we do gets transformed, and if it's successful, the world moves out far beyond it. Fred Sanger in England and I in the States found ways of working out DNA sequence very easily and very rapidly. And as I said, the problem before that was one of considerable difficulty. We could work out 1 base a month with great effort. But in any case, we had worked out that first piece of sequence. Then we discover very rapid methods. And those methods get used, we give them away. And they begin to get used around the world in '76. By '86, about 8 million bases of DNA have been sequenced everywhere in the world. By '96, it's 800 to 1 billion. And by 2000, it's already the human genome of 3 billion.
Yeah.
The process went very, very rapidly.
Yeah.
The chemical method, which I and Alan Maxam developed, was used for about 10 years, and then as the machines came in, Fred's method began to be used entirely, but by now the machines have gone far beyond that.
Yeah.
I mean, the first human genome took about in actuality about $600 million to sequence, and 5 to 7 years in many laboratories, and the machines now will do a human genome in about 30 minutes. And the cost is now somewhere probably about $200 to $300.
It's amazing.
And here, of course, is Dr. Gilbert winning the Nobel Prize in 1980 for that work. So now we're going to talk a bit about the dawn of molecular biology. And for those of you who haven't read it, I strongly encourage that you read a book called Invisible Frontiers by Stephen Hall. So we've gone from purifying insulin to understanding its amino acid structure and then cloning its gene. But this was the dawn of the era of molecular biology. Now DNA sequencing and the genetic code were understood. People had developed restriction endonucleases, although early scientists would have to create these themselves, purify them from bacteria, the fact that there are specific enzymes that will cut DNA at specific sequences. Synthetic organic biochemistry was in its infancy, where you could actually create nucleotides. The ability to clone messages and reverse transcribe messages of mRNA into complementary DNA, and vectors for carrying those genes in and out of bacteria and eukaryotic cells was all developed in the 70s, such that it became possible to clone things like human insulin. And we take it for granted now, but that field of science was highly controversial. When it was first announced in the press that the scientists at Harvard were going to clone human insulin, here's Mayor Vellucci, the mayor of Cambridge, had public hearings where large numbers of people would show up to protest this type of science. Here's a letter that he wrote to Dr. Philip Handler, who was then the president of the National Academy of Sciences, in which he described a man in New Hampshire seeing a hairy 9-foot creature that they thought may have been a consequence of these genomic engineering studies. And here's some other comics from that era, from the 70s, that were shared with me by Professor Witkowski at Cold Spring Harbor Labs. Here we see Run for the Hills, the recombinant DNA has escaped. And here we see a mad scientist running in with Frankenstein and all of these creatures, Alice in Wonderland, et cetera. So that the idea of cloning human genes was quite controversial. At scientific meetings, people would show up carrying banners like this across the stage. So that was the setting. And then in May 1976, Eli Lilly convened a meeting in Indianapolis, Indiana, because their experts had observed that the prevalence of diabetes was increasing, and they projected that the supply of beef and pork insulin was finite. So Irving Johnson, the Lilly vice president at that meeting, said it was clear that if something didn't change, a shortage of insulin would develop. Now, at that meeting, there were scientists from San Francisco, Southern California, Harvard, and elsewhere, leading scientists in the field of molecular biology. The San Francisco team was led by Professor Bill Rutter and Howard Goodman, and young scientists there at the time, Axel Ulrich, Graham Bell, and Herb Boyer, who along with Stan Cohen is credited with recombinant DNA technologies, were part of the UCSF team, all brilliant. The Harvard team, we've already heard from Walter Gilbert, Arj Efstratiadis, and Lydia Villa-Kumarov, and others were there of the Harvard team. And then there was a third team at the City of Hope, and I'll describe them more in a moment. But they all left Indianapolis with the race to be the first to clone human insulin so that bacteria could be trained to make human insulin to overcome this projected shortage of beef and pork insulin. So let's hear from Dr. Gilbert again, describing the political climate of the era.
What can you reflect any on Jeremy Rifkin and the impact that had on your work and the political atmosphere?
is there a conceptual possible difficulties with this idea of cloning genes and moving pieces of organisms around?
Right.
And so I think it's actually in the Gordon conference in '74 that we write a letter to NIH saying we should look at the government should think about, is this a problem?
Right.
And shortly after that, we actually get federal guidelines as to how to do the recombinant DNA research. And so one line of scientists is thinking, yes, we're using safe bacteria, we're taking cautions in how we do the research, we're using laboratories, special laboratories if we need them, but one side of the scientific community at Harvard began to argue against it. And that argument first was in front of the dean, a curious moment in which Ruth Wald talks about how unknown and dangerous the science is, and you shouldn't explore the unknown. And the dean says, but I thought the point of science was to explore the unknown.
So here's the third team at the City of Hope. This is Arthur Riggs, Kiichi Itakura, Dave Goodell, and Roberto Crea. They took a different approach. The San Francisco team and the Harvard team were using molecular biology of cloning the messenger RNA and then from that creating cDNA. The City of Hope team decided to try to create an artificial insulin cDNA by synthetic biochemistry. Here we see these people were young once. Here's Art Riggs. This is Herb Boyer, Kiichi Itakura, Roberto Crea, Herb Heinecker, and some of their team there at City of Hope. They put in a grant saying, we think we can make these genes and we can put these genes into bacteria and that they'll make proteins. They proposed making somatostatin first because it's small and simple, and then subsequently insulin. And for those of you who've had study critiques that haven't met with your expectations, I want you to read these words. They said, in conclusion, the goals reflect extremely complex and time-consuming projects which may not be reasonably accomplished in 3 years. The only possible outcome of this work would be to confirm that these manipulations can lead to the synthesis of a human peptide in E. coli. Because of the poor choice of biological system, this appears to be an academic exercise, and the grant was not funded. Nevertheless, they persisted. That's one of the themes I wanted to cite from the beginning. And here's a handwritten note from Bob Swanson, the co-founder of Genentech, and their patent lawyer, Tom Kiley, to Art Riggs, showing, because the first protein peptide that was made in bacteria was somatostatin at Art Riggs' insistence. And what Bob Swanson wrote was, everything that Genentech is and will be began with this. Thank you. Again, Art Riggs described sitting by the scintillation counter looking for evidence that somatostatin had been made by bacteria. And first time they tried it, it didn't work. And Bob Swanson had to go home, and he was thinking he was having a heart attack because they'd invested so much in this. But second time it did work. Well, I told you that that meeting in Indianapolis was in 1976. By 1978, the team at City of Hope had succeeded in getting E. coli to make human insulin. And here's more recent pictures of Art, Dave Goodell, Herb Heinecker, and Kiichi Itakura. This, again, was a shot heard around the world. Because once you have insulin cloned, then you can modify it. So that we now, and we take this for granted, we have all of our various designer insulins in addition to systems for infusing insulin, the continuous insulin infusion systems, pumps as we call them, and CGM systems, such that now the prognosis of diabetes has continued to improve. So what was the significance of this insulin somatostatin project? They were the first man-made and designed and made genes made in bacteria. They demonstrated the power of recombinant DNA technology combined with chemical DNA synthesis. They were the first FDA-approved recombinant DNA-derived biologics. And it led to the launch of Genentech and Biogen and Amgen and all the other large biotech companies that you know of today can all trace themselves back to somatostatin made in bacteria there at the City of Hope. Time magazine in 1979, I've showed you earlier pictures of Herb Boyer, but the tipping point occurred with insulin. When insulin was made in bacteria, people realized this new scientific field was for real. So I asked Dr. Gilbert to comment on that as well.
Just reflect for us the impact of that era on modern medicine.
Is the development of the biotechnology industry. And in fact, the entire effort to make first useful human genes in bacteria or in other organisms. That's produced a wave of pharmaceuticals that have had a great impact on people. Mm-hmm. The entire development of antibodies as made then in cells, mm-hmm. and then used as drugs are still a dominant pattern of drugs today. Mm-hmm. Small interfering RNA molecules have become drugs. Mm-hmm. The drawback with the protein drugs, just like the drawback with insulin, was that it was something that you had to inject. Mm-hmm. And you couldn't take it by mouth, and so that the question of whether you can have an oral drug is still a dominant one. Mm-hmm. There's still an effort, I think, with insulin to get it into the body in other forms.
Yes, sir.
And then I assume that there's some young scientists on this conference, so I asked Dr. Gilbert for the Levine Symposium to render his advice to the young scientists in the crowd.
The Levine Symposium is well attended by junior scientists. Any reflections for them? Any comments to them that you'd like to share?
Well, I think science is some of the greatest fun you can have, is doing science. As scientists, we're constantly interested in the latest thing. Yeah. Sometimes it's useful to know a little past history, past knowledge. I think the best advice is the one you just quoted to me, which is actually find good projects, work on things that have significant outcomes.
Anna mentioned when she introduced me, I was privileged to help organize a meeting to honor the first production of human insulin and bacteria at the City of Hope a little less than a year ago. And in attendance that night, here's Art Riggs. Dr. Riggs yesterday donated $100 million to the City of Hope. The royalties from the patents of the work that I described have been good financially for him and the institution. But he's a gentleman and a gentle, great scientist. Here's his technician, Louise Shively, and her husband, Jack. Here I am. Fouad Kandil, who currently heads the diabetes clinical efforts at City of Hope and research efforts. Here's Graham Bell today. He was part of the UCSF team and is now at University of Chicago. Herb Heinecker, Lydia Via-Kumarov, and her husband, Tony, who were part of the Gilbert team. And here's Stephen Hall, who wrote that great book, Invisible Frontiers. This was a thrill for me. When I was able to introduce them, I said it felt like I was calling John, Paul, George, and Ringo to the stage. These are all scientific superstars. So what have I touched on today? The major themes I told you were good ideas versus brilliance. Sanger said he wasn't academically brilliant, and no one ever accused Banting of being brilliant, but they had good ideas and they were persistent, and they succeeded in bringing their idea to fruition. I talk about scientific progress and the pace of it. It seems very slow for a long time, but then if you get on the right idea, it can move very fast. In Banting's case, he went from his middle of the night dream to injecting the first dose of insulin within 13 months of each other. And the Indianapolis meeting, identifying the need to create human insulin and then having it done within 2 years, is another example. Serendipity plays a great role in science. Banting happening to read Moses Barron's article the day that he'd given this pancreatic anatomy lecture. The observation that Burson and Yalow made about insulin's circulating half-life in people who'd been injected with it earlier. And Dorothy Hodgkin seeing insulin crystals as a young scientist and devoting a 35-year career to being able to figure out insulin structure. We've talked about the importance of persistence and overcoming hardship. This is especially important for the young scientists in the crowd. I've told you that Burson was rejected from 109 medical schools before it was accepted, and they were their paper on antibodies was rejected. I've told you that Dorothy Hodgkin had to work for 35 years to achieve her scientific dream. And certainly Banting and Best had a miserable summer that summer before they succeeded in some evidence that they had isolated insulin. And then some of the unforeseen consequences of observation, careful observation that I had mentioned. Yalow and Burson's prolonged insulin half-life, leading to radioimmunoassays, which essentially opened up the field of endocrinology. Where would we be if we couldn't measure the hormone levels? Sanger and Alan Maxim figuring out how to sequence DNA and RNA, and then the observation of restriction endonucleases allowing recombinant DNA technology and the designer insulins. And then, of course, the molecular biology revolution and biotechnology that we take for granted today. So early in my talk, I shared that the last paragraph of Banting's Nobel lecture begins with the sentence, "Insulin is not a cure for diabetes." He goes on to say, "It is a treatment. It enables the diabetic to burn sufficient calories so that proteins and fats may be added to the diet in sufficient quantities to provide energy for the economic burdens of life." Just outside his home, which if this wasn't here, you'd see his home where that dream occurred, and his clinic, by the way, was the first floor of that home. His bedroom was on the second floor, is a flame, and you can't see it. It's not an eternal flame. It says it's the flame of hope. It was lit by Her Majesty Queen Elizabeth, the Queen Mother, on July 7th, 1980, not to burn forever. And then the team credited with finally curing type 1 diabetes gets to extinguish that flame. I hope that through work that we do and is done at Stanford and by many of you who may be listening to this lecture, that I live long enough to see that flame extinguished. And with that, I'll close.
That was fantastic and every bit as good as I remember from a year ago. What a treat to hear the science, to see the photographs of the people who have contributed so much to the field. And I totally get your points here about sharing with the younger generation, and I count myself within that, about the need for persistence, the role serendipity plays within our research. So important, because it can become so easy to focus on the successes and not appreciate the luck and the battles that have gone on in the background. I was going to ask you about comments on team science. I know that the start, when you told us about Banting, Best, Collip and McLeod, you alluded to some of the troubles that were happening within that team, which I think are understandable. But clearly it was the teamwork there that brought around the success, bringing together diverse skill sets with different ideas. And I wondered if you could comment a little bit on how you see the role of team science in these major achievements.
I failed to call attention to it, but what you're alluding to, Anna, is the infighting in Toronto was terribly bad. When Banting received the call that he had won the Nobel Prize, initially he said, "Oh, that's great." And then they said, "Yes, we'd like to invite you and Dr. McLeod to Sweden to receive it." And he said, "McLeod? If McLeod's getting this with me, then I don't want it." And he refused it. And later people said, you can't refuse a Nobel Prize. And so Banting said, well, then I'll share my half with Best, because he's the one that did the work with me. And when McLeod heard that, well, he said, well, then I'm going to share my half with Collip, because he's the one that really purified it. So I'm highlighting the acrimony that can happen with success, and it happens frequently with success. But it's also true that each one of those scientists played an absolutely integral role in the purification and first use of insulin. But really, if it wasn't for Collip crystallizing insulin, they never would have been able to treat Leonard Thompson. And if it wasn't for McLeod's insights into how to conduct the experiments, they wouldn't have succeeded. So each of those four had important roles to play. I cite this quote by Louis Barker, who was the chairman of medicine at Johns Hopkins, or a professor of medicine at Johns Hopkins, but he visited Toronto shortly after insulin was purified, and he witnessed some of this infighting. And his advice to them all is, you guys got to stop this. And he said, in insulin, there is glory enough for all. So there's a wonderful movie that you can watch called Glory Enough for All that very accurately tells the story of insulin's first purification and use. It's wonderfully done. I'd recommend anybody to watch it. So yes, each one of those people played a very important role. Group science is very important, borrowing on each individual's expertise, but acrimony can oftentimes accompany such success.
And I think a slightly related question, and one that I think is also relevant, thinking about how we conduct science today, is the role the combination of academia, biotech and pharma have played in propelling the success of this story. So right at the start, you talked about Eli Lilly, but of course there's also Novo, and thinking about the fact that this wasn't something that just happened within an academic setting. Very quickly and rapidly, as you described, pharma were brought in, and then more later in the journey, the biotech industry. And I guess that's still a really important model for how we think about scientific discovery and translation in today's setting. But was that particularly unusual at the time?
Well, I'm glad. Actually, Anna, the next time I give this talk, I'm going to throw that in as another kind of unforeseen consequence. It was very unusual at the time. Those of us with gray hair recall when in the 70s, when any scientist that went from academia to industry was considered to be going to the dark side. You didn't interact with industry. In fact, Banting sold the license to insulin for $1. He didn't want to make it. Was considered beneath a scientist to be involved in the business of medicine. Similarly, the scientists at Mayo Clinic that purified cortisone didn't patent that. So the relationship between academia and industry is a relatively new thing since the biotech industry blossomed in the 70s and early 80s. So yes, I in retrospect, I should add that that's another way that insulin led the way by very early on in involving commercial companies in its purification and distribution.
David, I'd like to thank you again for a really fantastic lecture and such a fitting start to today's symposium.